Welcome to the webinar on elemental microstructural analysis. My name is René de Clou. I'm with EDAX in the Netherlands, and I'll be talking in the first part of the webinar on EBSD and EDS in the scanning electron microscope.
And my name is Katie Rice at CAMECA Instruments in Madison, Wisconsin. We are a sister company with EDAX, and I'll follow René's talk with a little bit about atom probe tomography and how that can be combined with transmission EBSD specifically for a wide range of microstructural analysis capability.
This is a short outline of the talk. First, what can you actually do with combined EDS EBSD data? It is often claimed that it's only useful to distinguish phases with similar crystal structures or EBSD patterns. But there are some more instances where having EDS information available can be a great help. Then I will briefly review Chl-scan, the patented approach to assign EBSD structure files to specific areas in the scanning field, before going into some physical limitations regarding the interaction volume differences between EDS and EBSD and what can be done about it. Another often accepted limitation is that you will typically have relatively high beam currents, which requires the use of very short EDS pulse processing times, which then in turn limit the detection of low energy X-rays. When we then zoom into a higher resolution analysis, we can go away from doing EBSD on bulk samples to use TEM foils for transmission EBSD. And when even higher resolution is required, EBSD can be paired with atom probe tomography, which will be presented by Katie Rice. Combining EDS and EBSD mapping is not new. This is the oldest example that I'm aware of from 1997, and as you can see from the EDS map on the right, the high nickel content nicely matches the small particles that you can recognize in the SEM image on the left. So there's a direct correlation between the chemistry and the microstructure. But this was 20 years ago. Things have changed a lot since those days. Now you can routinely collect EDS data with almost all EBSD scanning modes. This slide shows you an example of a combined scan on a geological thin section of an olivine orthopyroxene rock. So this was a combined beam stage scan collected over 435 fields, which is almost 3.5 by 1.5 centimeter in size. During acquisition, I only used the matrix phase, the red phase that you can see here, that's the olivine, and we collected the chemical information to allow subsequent processing and adding the missing phases offline. Similarly, you can apply the same type of multi-field mapping to the third dimension. So instead of moving the stage and do another beam scan, you can actually go to a FIB-SEM microscope and do sequential slicing of your sample to collect a three-dimensional volume. And during each slice, you can actually collect a combined EDS EBSD map. In this case, it does not only allow offline verification of the phase assignment, as we saw in the previous slide, it also allows the analysis of the chemical composition inside grains and along grain boundaries. So if I pull out one of the grains here on the left, you can see that the chromium concentration does not match the grain outline and is most likely representing an earlier state of the material. So why would you start collecting EDS and EBSD at the same time? One of the reasons, as you see here, is differentiation of phases that produce very similar EBSD patterns. This example is from a calcite dolomite rock, and as you can see in the image in the middle, the red and green, that doesn't look very realistic. The EDS maps on the right, the top is calcium, the bottom one is magnesium, which corresponds to the dolomite phase, show the proper phase assignment. When you now can use that information into the indexing process, you can teach the system when to use which structure file, and that can actually give you the proper phase assignment and good analysis. So here in the middle, we now see the reconstructed phase map, and on the right hand side, the top, you can see the calcite matrix, and at the bottom we have the IPF map of the dolomite phase. But materials that exhibit similar patterns are not the only case where EDS information can help a lot. A notorious problem is often the difference in polishing response of different phases in a multiphase material. So even when the polishing is optimized for the matrix material, quite often other phases in the sample do not respond in the same way to the polishing procedure. As you can see in this image here, on the left hand side, The dark areas, that's where a phase used to be that was almost edged out during the polishing process. On the right hand side in the phase map, you can see that in some places there is still a little bit of magenta left over of that phase. When you have the EDS information recorded as well, the EDS can tell you what the surface area of that phase is and ensure that the area fraction is properly accounted for. And finally, EDS is indispensable in materials with lots of phases. Without EDS, each pattern would need to be matched against all candidate phases, as you can see in this iron silicide material, in which 8 phases are recognized. Matching all candidates to all patterns may work well when the pattern quality is very high and they all have a different structure, but it might be slow. Another issue occurs in areas where the pattern quality is not so good or at grain boundaries, and that will typically cause misindexing and wrong phase assignments. One of the best ways of recording such a scan would be to run it with only a few main phases, maybe 1 to 3, and make sure that you collect all the EDS information you need to identify the missing phases later offline. That is a much more efficient use of SCM time, and all omitted phases can then be added offline afterwards. Now, how does that actually work? All EDAX EBSD indexing software offers the Chl-scan functionality, or chemistry-assisted indexing. The key to this operation is the direct correlation between the patterns and EDS signal at each pixel. In the example on this slide, you can see that the titanium matrix is identified properly, but inside the larger grains inside the crack, the yellow aluminium oxide has been misidentified for titanium in certain places. Now, note that the twin lamellae are correctly identified, even when the main body of the grain appears speckled. This is caused by orientation-specific pseudosymmetry. In some cases, EBSD patterns of different phases, even with different crystal structure, may appear very similar for certain orientations, while other orientations are so different that phase discrimination is not a problem. The phase assignment using chemistry can either be done automatically or manually using interactive highlighting. In this slide, you can see 2 EDS charts or frequency histograms that illustrate the number of EDS counts at all pixels in the maps. By selecting the high signal parts of both charts, you can easily select the target grains in the map. These colored points are then assigned to the desired phase for reindexing. This chemical verification then prevents misindexing or a phase mix-up at certain orientations, even if the patterns happen to be very similar. The final phase assignment is now correct, and the dataset can be analyzed further, for example, by looking at the grain size distribution for each phase individually, or you can investigate the misorientation distribution along the phase boundaries. Now let's take a look at the interaction volume. The typical information depth for EBSD patterns is somewhere between 10 and 50 nanometer, depending on the atomic number of the material. At the same beam conditions of 15 to 30 kV, the EDS source volume is easily 1 micron. This causes a mixed signal in the EDS spectrum at a phase boundary, even when the EBSD pattern is produced in only 1 grain. This can cause trouble along these boundaries when you are scanning a material with step sizes much smaller than the EDS interaction volume. In this example, it only causes the boundaries between the pink and blue grains to appear a little fuzzy. Now, if you want to sharpen this up, there is a tool available in the software that allows you to average all the EDS counts inside each grain as identified by the EBSD orientations. And when you do that, the EDS information and also the phase boundaries actually get really sharp around the orientation change or around the phase boundaries. This is especially useful if phases have very similar crystal structures and can be indexed as either phase, and only the EDS shows the difference. When we zoom in a little closer, you can see that the EDS signal gets a really sharp transition right at the grain boundary. On the left side is the original EDS map, where the grain edges are not quite sharp. Using this chemical information could cause a small shift in the phase assignment, and that can result in a 1 or 2 pixel shift of the grain boundary in the EBSD map. When the phase assignment does not actually coincide with the grain boundary, that will affect your misorientation distribution analysis. The map on the right shows the EDS after applying the average EDS data tool. And now the chemical boundary and the phase boundaries match precisely. In this example, the effect is perhaps limited, but when we look at the material with smaller particles, the effect becomes very noticeable. You can see that in this map of a nickel alloy with chromium carbides that was collected with a step size of 100 nanometers, which is much smaller than the EDS interaction volume. The entire map in this case was indexed using the nickel structure file alone, which is face-centered cubic and also indexes the carbide grains very well. The resulting grain structure from this indexing is superimposed on the chromium EDS map on the right, and you can see that the EDS signal of the carbide grains clearly extends across the indicated boundaries well into the adjacent nickel grains. Zooming in shows the effect more clearly. The EDS signal suggests that the carbide grains are larger than they really are. The true size can be seen in the IPF map on the right that was indexed with the nickel structure file only. Here the EBSD indexing shows the actual extent of the carbide grains. When you would now re-index the scan file using Chl-scan and the original chromium signal, this is what the phase map would look like. All carbide grains in green now have a ring of pixels around them that actually belongs to the nickel matrix. This would not only affect the grain size determination, but calculating a misorientation distribution from such a map would actually use orientations that are fully inside the nickel grains, which makes the misorientation distribution between the carbides and the nickel matrix invalid. After applying the EDS averaging inside each grain, the EDS map now displays the true carbide grain size, and also the reconstructed phase map is now correct. Now let's take a step back. Another typical issue is the scan speed when you are collecting EDS and EBSD simultaneously. Often the scan speed is reduced on purpose to allow more EDS counts to be collected or to ensure that also a strong signal is obtained from light elements such as carbon and nitrogen. The EDS maps on this slide show the chemical contrast that is obtained when the maximum number of counts in a single pixel would be 100, 300 or 1000 counts. With 20,000 counts input, that would dictate scanning speeds of 200, 66 or just 20 points per second. Increasing the count rate to 60,000 counts effectively triples the speeds that may be used to get the same quality EDS maps. In the past, typical collection speeds were limited to 10 to 50 points per second. Any higher speeds would require much higher EDS input count rates, which would then need short pulse processing times, which made detection of low energy X-rays impossible. Recent improvements of EDS detector technology has overcome this problem. The latest EDAX Elite detectors can easily process more than 750,000 input counts and then produce output count rates exceeding 400,000, while still detecting, for example, carbon. This either allows for very fast combined EDS EBSD mapping or for greatly improved counting statistics. Here is an example of a combined EDS EBSD map on a ferritic material with chromium carbides. The simultaneous EDS data was collected with an input EDS count rate of 750,000 counts per second and a throughput count rate of 420,000 counts per second using an Elite detector. When EDS spectra are then extracted from this dataset afterwards, changes in carbon content of only a few weight % can be shown between the matrix and carbide grains. The patterns from the EBSD then confirm the type of carbide as a chromium 3 carbon. Here you see side by side the ferrite chromium carbide phase map on the left and a blended iron carbon EDS map on the right. And note that even the smaller submicron carbide particles are clearly visible in the carbon signal collected at these extreme high count rates. One of the things that limits the size of features that may be analyzed with EDS is the interaction volume, where X-rays are generated in the bulk of the sample. An efficient way to reduce this interaction volume is to take the bulk of the sample away and just work on a thin electron transparent foil. This also allows the EBSD data to be collected in a different configuration that allows collecting maps with high spatial resolution. For this, the sample has to be mounted close to horizontal under the pole piece. The patterns are then generated at the bottom of the foil and projected onto your standard EBSD detector. No special detector modifications or software settings are required apart from a dedicated specimen holder. The chamber view images here show the measurement geometry with two different transmission EBSD sample holders combined with a Hikari EBSD detector in a Joule 7100F and an FEI Quanta 250 FEG scanning electron microscope. In many cases, the space close to the pole piece in the electron microscope is very limited, and mounting the foil horizontally could actually cause parts of the EBSD phosphor screen to be in the shadow of the sample holder, as you can see here on the EBSD pattern on the left. This can be prevented by tilting the sample a little away from the EBSD detector. And as an added advantage, now also the solid angle of the EBSD pattern increases, which simplifies consistent indexing. Furthermore, the intensity gradient and band size variations from top to bottom on the phosphor screen are minimized, which also makes the analysis easier and faster. An argument to keep the sample horizontal would be to minimize the beam cross-section where it hits the specimen. But tilting the sample a little bit does not affect that too much, as you can see in this chart. The effect of the specimen tilt up to about 40 degrees is very limited. Here is a nice example of the EBSD mapping resolution that can be obtained in a transmission EBSD configuration. This data was collected by Natasha Erdmann at Joule in the USA using an EDAX-Hikari EBSD detector. The map on a fine-grained gold film was collected with steps of only 3 nanometer, which allows successful orientation and grain analysis of grains that are smaller than 20 nanometer. In this second example on a silver foil, some very thin twin lamellae down to 5 to 10 nanometer can be observed. Note that mapping such small details would not be possible in a bulk specimen due to the larger interaction volume. Transmission EBSD can also be very useful at lower magnifications, where you can analyze the entire electron transparent area of your foil before going to the TEM for further analysis. The area where patterns can be successfully analyzed is limited by the foil thickness. As the sample gets thicker, the pattern contrast rapidly decreases until indexing becomes impossible, as you can see in this aluminium-magnesium alloy. A new processing tool, NPAR, which stands for Neighbor Pattern Averaging and Reindexing, has recently been introduced in the TEAM and OEM Analysis 8 software packages. It allows for offline averaging of all surrounding patterns onto each pixel during reindexing. This averaging strongly improves the pattern signal-to-noise ratio, which allows successful band detection and indexing over a much larger area than previously possible. When you remove all the points that it could not be confidently indexed with a single orientation solution, the effect becomes very clear. When you zoom into a section at the top left of the sample, small particles that did not index properly become visible in the IPF map. The intensity in the simultaneously collected aluminium EDS map strongly changes with the specimen thickness, but the sample also here clearly is not homogeneous. The location of small particles can be seen by the reduction of the matrix aluminium signal. The simultaneous EDS mapping was collected as a spectral dataset, where the EDS spectra are recorded at every pixel in the scan. This allows the user to extract EDS maps of elements that may have been missed during the initial mapping. Here you can see the separate iron and silicon maps of the analysis area shown in the previous slide. The iron-rich particles appear in trails, while a few larger silicon-rich particles are scattered throughout the sample. Now let's take a close look at the silicon particle that sits here in the thinnest part of the sample, right at the edge of the hole. At first sight, the particle appears to be clearly different from the matrix. It is brighter in a transmission four-scatter detector image on the left, and this corresponds nicely to the silicon EDS map on the top right, which is superimposed on a Prius image showing beautiful orientation contrast. This image is generated by collecting the intensity of the electron signal in the middle of the EBSD phosphor screen at each pixel. However, there is no orientation change visible when you go into the particle when you look at the IPF map on the lower right. This lack of orientation change is confirmed when you look at the individual EBSD patterns. The Prius center map and the IPF map show strong orientation contrast variations around the particle. But the individual patterns across the profile show that this external deformation is even stronger inside the particle. So the crystal appears to be epitaxial, but with a significant misfit of the lattice. So the sequence of patterns shows that the bands are continuous across the grain, but only the 111 and 220 bands remain clearly recognizable. The other bands are getting very blurred due to extreme dislocation densities. The iron-rich particles are different. These grains are smaller and they actually do produce discrete patterns, but getting these patterns is very difficult due to interference with the over or underlying aluminium lattice. In many cases, the iron particles are so small that they don't continue from the top to the bottom of the foil, and you always have a little bit of a signal from the aluminium matrix that overlies the patterns. When all the patterns are recorded during mapping, the user can easily browse through all these patterns by just moving the mouse over all the particles that are visible in the map in OAM analysis. This way you can judge the quality of all the patterns that are available, such that you can select the best pattern to be used for the phase identification. In general, it is also a good idea to collect a reference pattern of a known material nearby, in this case the aluminium matrix, to verify that the calibration is set as good as possible when you are doing the phase identification offline, away from the microscope. Now, in this case, the original pattern has quite a poor signal-to-noise ratio, but also here that can be improved with a local NPAR averaging followed by a contrast brightness optimization. Phase identification is then done by carefully identifying which elements have to be coming from the particle and which elements might actually come from the over or underlying aluminium-magnesium matrix. In this case, the pattern indexing result was a close match with a monoclinic aluminium-13 iron 4 phase. To summarize, EDS and EBSD can be collected and used together over a wide range of scales, from the centimeter to the nanometer scale, with the nanometer scale analysis performed in transmission EBSD mode. When even higher magnifications or detail are required, another analysis system is needed. The second half of the webinar will be presented by Chr₂ Rice from CAMECA and illustrates the use of EBSD to assist in the preparation and analysis of atom probe tomography data.
Hi, as Renee mentioned, my name is Chr₂ Rice. I am an application scientist at CAMECA Instruments in Madison, Wisconsin, and I'd like to introduce a technique that's really complementary with the techniques of EDS and EBSD that Renee has already gone over. So atom probe tomography is really the ultimate tool for nanoscale compositional characterization. We're detecting individual atoms and positioning them with near atomic spatial resolution in X, Y, and Z. We're also detecting their elemental identification by time-of-flight mass spectroscopy. So by combining these two pieces of information about their 3D position and their elemental identity by mass spec, that will enable us to create a 3D model of any kind of compositional variations within the specimen. So I'd just like to spend a couple minutes talking about how this technique works. As I mentioned, atom probe combines projection imaging with time-of-flight mass spectroscopy. So in order to get this information, we start with a needle-shaped specimen of a radius between usually 50 and 100 nanometers. We pulse a field on the surface of the tip by either doing a voltage pulse or hitting the tip with the laser, and that would be a thermal pulse. So when this process happens, a single ion flies from the tip to the position-sensitive detector. By measuring the time it takes for that process to happen, we have a time-of-flight measurement. And by looking at the position on the detector, we can tell where that ion came from on the tip. So by combining these two pieces of information, we can create a 3D model of the ions in the specimen, up to 80%. And we're able to apply a variety of different analyses on that model and look at things like local chemical variations or grain boundary segregation or layer thicknesses and so on. So these two pieces of information are very powerful in allowing us to tell a lot about our material. So in terms of applications, really any conductive, non-porous sample is appropriate for atom probe tomography. We can look at things like semiconductor devices where we have local variations in dopant concentration. Traditionally, metallurgy has been a very large applications area with things like looking at clustering and steels, and even ceramics and other types of oxides. So I'd like to take the rest of this talk and really focus on the problem of grain boundary segregation because that's very complementary to the techniques that Renee's already discussed. So the first step in the sample preparation process for atom probe is to actually find an ROI that we care about. And one way to do this in the case of grain boundaries is to take a reflection EBSD map of a highly polished sample and process the map with OIM analysis to look for certain types of grain boundaries. We can then use a focused ion beam or a dual beam microscope to continue the preparation process so that those samples are suitable for atom probe, which I'll describe in the next slides. So once we've identified the grain boundary of interest from our reflection EBSD map, we can begin the process of preparing this into a needle shape that's suitable for atom probe. And the way this works is by using a focused ion beam microscope to target a specific region of interest. So first we can place a protective strip of platinum over the region of interest. We can remove material around 3 sides of the region by angled cuts to produce a long wedge of material with the ROI. We can then bring in a micromanipulator such as an OmniProbe or Easy Lift and weld the wedge to that needle and cut it free from the bulk. We can then drive the wedge over to a flat-top silicon post, which we call microtip coupons, weld the wedge to the microtip coupon, and cut a slice of that free from the longer wedge, and then weld the other side so that we're left with a tiny wedge of material on a flat-top post. So once we have a tiny wedge of material on a flat-top post, we can begin the process of sharpening this into a needle shape so that it's ready to go into the atom probe. And the method that we'll use is with a 30 kilovolt gallium ion beam, and we'll put a pattern over the top of that wedge with increasingly smaller inner diameter circles so that we slowly mill away the excess material until we're left with a needle. Now this process does tend to damage the surface a little bit and implant gallium ions into the material. So to minimize that, we'll finally mill with a 5 kV ion beam, which can get rid of most of the gallium ion damage. So one of the problems with the sharpening process is that although we may be able to easily see the grain boundary in the initial mounted wedge, we often lose enough contrast to be able to see it in the final tip shape. So that can be due to reasons such as gallium damage at the surface or simply because of decreased interaction volume available for imaging as the sample gets smaller and smaller. And this is where transmission EBSD can help us out a lot by enabling us to take successive maps at each stage of this milling procedure and be able to adjust our mill pattern so that we can position the grain boundary close to the apex and ensure that we collect that within our atom probe analysis. And the EBSD will also provide us information about the nature of the grain boundary, such as the misorientation between the two grains. So as I think Renee mentioned, no special equipment is required for transmission EBSD beyond a special sample holder that will allow the beam to pass through the sample and be collected by our EBSD camera. And luckily for atom probe, we have several different options in what types of samples to use and how we can mount those in our microscope. So most often at CAMECA, I'm using a micropost array, which can accommodate either 22 or 36 atom probe samples on a small piece of silicon. And when I'm working with those, I like to mount them to a pre-tilted holder, which will allow me to get a little bit better tilt in our FEI microscope. We can also use wire geometries or half-grid geometries, and we make special holders at CAMECA for those. And the advantage of the half-grid is that we can then do TEM on these samples And all of these are designed to go directly into the atom probe after transmission EBSD or other correlative microscopy. So EDAX has really made this process easy for us by introducing a new mode in their team software called Atom Probe Assist, which has several different features that can really help you target a grain boundary during the fib sharpening process. So I'd just like to talk about a few of those features and how I use them, and hopefully you can implement them in your own lab. So one thing that they've added is an automated angle correction specific to transmission EBSD. So the transmission EBSD angle is calculated from horizontal, and that's a little bit different from how reflection EBSD angles are usually reported. So our ion column in our microscope is at a tilt angle from vertical of 52 degrees. So when I'm sharpening a sample that's normal to the ion beam, my EBSD angle will be 90 minus 52, or 38 degrees. So the first thing I do is check that box and enter my 38-degree angle there. Secondly, they've added an optimization button for the camera settings, which will allow the computer to choose the optimum camera settings, such as gain and exposure, for you. So I like to put the cursor kind of in the center of my conically shaped atom probe specimen and then click Optimize, which will allow the computer to choose what it believes to be the best camera settings. And sometimes I adjust them, but I usually find it works pretty well. So included within this Atom Probe Assist optimization settings is a background subtraction recipe that it automatically applies, which was developed by Renee. So this includes a dynamic background subtraction, which helps when going over the varying thicknesses and vacuum that you might encounter by taking a map, an intensity histogram normalization, and a median smoothing filter, which can help you, the software, identify bands. So typical microscope settings that I usually use are around 30 kV and somewhere between 0.5 and 1 nanoamp of beam current, but I have seen samples like oxides where 20 kV might work better. So this is going to be pretty sample dependent, and you might need to play around with those to find the optimum settings. So one of the more common questions that I get is how to minimize contamination for atom probe samples when mapping them using transmission EBSD. And this is a pretty serious concern because that can affect your ability to run these samples in the atom probe. So I've come up with a list of a few tips that you may want to try if you're struggling with contamination on your samples. So one thing I like to do is use survey mode whenever possible over point analysis mode. That way the beam is not sitting on the sample for a long period of time. You might want to also try using the auto beam off option after the map is complete, so you're not unnecessarily exposing the sample to the beam. The other thing I like to do is adjust camera parameters of gain and exposure when the beam is not exactly on the tip. So what I like to do is quickly look at the Chr₂ C patterns, and then if I need to make an adjustment, I'll use survey mode to move the beam off the tip, make my adjustments, and then check them by moving the beam over the tip again. One thing that might help is using a larger step size, lower exposure times, and higher accelerating voltages. And probably the most common tip is, or the most important tip, is to decide when you need to map and when you don't need to map. So maybe survey mode is sufficient to see the position of the grain boundary within your sample, and you don't need to collect a full map. Of course, for publication purposes, you may want to have a nice map with a small step size, but again, that may contaminate your sample more. And of course, always keep your chamber clean by regular plasma treatments. So for the rest of the talk, I'd like to focus on some real-world examples of how we can combine transmission EBSD with atom probe tomography for a true nanoscale characterization of the specimens. Here I have a grain boundary in a boron-doped steel. Transmission EBSD has allowed me to get the grain boundary of interest at the very apex of the specimen. And in the center there, you can see the ion map of the atom probe tomography results. showing carbon and boron segregation at the grain boundary. So the question becomes, how can we actually quantify the amount of segregation for both boron and carbon at the grain boundary in a 3D way? So one way to do this is to take a 1D composition profile in a certain region of interest across the grain boundary. That will allow us to see an increase of carbon and boron across the interface, but I want to know more about this sample, and I want to know how we can get that segregation measurement in 3D. So one way to quantify segregation is using a technique called Gibbsian interfacial excess. So that's a measurement of the amount of segregation at a grain boundary normalized by area. And this amount of segregation is in excess of the amount that is found in the matrix. So what these measurements are going to allow us to do is calculate how much segregation is at an interface normalized by whatever sampling area that we're using. So in this case of boron-doped steel, I've taken the interfacial excess measurements of both carbon and boron, and you can see them in the table below. So one thing that these measurements can tell us is that if we see something very high, like the carbon, 34 atoms per nanometer squared, that will tell us that, this is really more than can be a monolayer. So that may indicate the presence of a separate carbide phase forming that EBSD really is unable to find. We can also look at the boron and see that that's much lower in the carbon and also unlikely to form a separate phase in this case. So our next question was, what can we expect to see at a low energy grain boundary such as a FCC recrystallization twin? So in order to find one of these, I took a reflection EBSD map of an Inconel 600 alloy and found a sigma 3 twin, which I can then measure the misorientation of, which should be 60 degrees. And these type of grain boundaries are energetically favorable for the material to make, so we wouldn't necessarily expect to see segregation there, but we weren't sure. So we had to take transmission EBSD maps as we sharpen the material to ensure that we capture the grain boundary right at the tip. When we looked at our individual ion maps of nickel, chrome, and iron, which are major components of the alloy, we didn't see any variations in composition or special segregation of those. And when we looked at our minor components of boron, carbon, and silicon, those were also uniformly distributed throughout the sampling volume. So to confirm that we've actually collected that grain boundary within our atom probe results, we took transmission EBSD before and after. I should mention that the sample did not fracture, so we were able to stop it before the sample fractured. And we can see that we did collect that second grain at the top here colored in red, so we can be reasonably sure that our grain boundary was captured within these ion maps here. So far, that's not a very exciting story of our FCC recrystallization twin, but Atom Probe can give us additional information about the field evaporation characteristics of this material. Here, I'm showing a density plot in atoms per nanometer cubed on the left, and we can see some changes in how this material evaporates based on which grain is evaporating. So in the first grain, we can see an area of high evaporation density where more ions might be evaporating at the pole. We can then see a discontinuity, which indicates our grain boundary, followed by an area where there's lower evaporation density at the pole. So evaporation field variations can occur in these pole regions, which can indicate changes in grain structure. Now that we can see our grain boundary, we can take composition measurements of those two grains. In this case, they happen to be very similar to each other, and although we didn't see any segregation, we can tell a little bit more about this specimen by measuring each grain individually. So lastly, I'd like to talk about what transmission EBSD and atom probe can tell us when we don't have any grain boundary in our specimen at all. And here I have a zircon geological sample. On the left you have an IPF map with an image quality overlay in grayscale. And the first thing I notice is a darker area of image quality near the center. Now if we look at our individual Chr₂ patterns, we can see a very sharp one at the tip, a slightly fuzzier one in the center, followed by a better one near the bottom of the tip. And the quality doesn't seem to be related to thickness variations in this case because our worst Chr₂ pattern is in a thinner region. If we look at our image quality only, we see a distinct area of low image quality in this blue region. So by taking atom probe tomography of this sample, we see that we have aluminum segregation at the surface of the sample. So because the Chr₂ patterns are coming from the exit surface of the specimen, this aluminum segregation at the surface may be disrupting the image quality of our Chr₂ patterns. So finally, we've gone over how we can prepare samples for atom probe tomography by using transmission EBSD and in particular atom probe assist. So our first step is usually to take a reflection EBSD map to identify any grain boundaries of interest. We can then lift out those desired grain boundaries and mount them to a substrate for sharpening. Next, we can start sharpening and thinning the sample and monitor the feature of interest with transmission EBSD as the sample gets thinner and thinner, and that will really enable us to target that grain boundary or other feature at the very apex of the tip. Thirdly, we can collect atom probe tomography information about these samples. Then we can take measurements such as the interfacial excess measurement to quantify segregation or any other kind of local chemical information that we'd like to get about this sample. So really the combination of these two techniques will give you a really good picture of what's going on in your material.
So let's summarize the webinar. As you have seen, EDS information can be combined with EBSD over a wide range of scales, from centimeters to atoms. And this EDS data not only facilitates discrimination of phases with very similar patterns, it also enables successful analysis of materials with up to 10 or even 15 phases. Special tools are available to minimize the physical effects of interaction volume differences, and recent improvements in EDS detector technology now allow extremely fast simultaneous EDS EBSD data collection, including the low energy X-ray lines. And when the analysis resolution in a bulk sample is not enough, TEM foils can be used to collect transmission EBSD data at a higher resolution. And when you need even more than that, you can go to atom probe tomography.
And atom probe tomography is really a complementary technique to the techniques that Renee has already discussed. EDAX has added several tools in their atom probe assist mode to help us in the FIB preparation of atom probe samples. Atom probe data can provide chemical information at the nanoscale, enabling local measurements of composition in 3 dimensions, and give us more information about grain boundary segregation. We hope you've enjoyed this webinar, and thank you to Renee for organizing it, and please let us know if you have any questions.